she said, this is I was comparing the parasite communities of the freshwater snail at Limiacama among tributaries of the upper Black Warrior Basin. Why is it interesting to study parasites in the snails is that it is novel. There's a huge diversity in snails. The highest diversity of Floricidae in the world is here in Alabama. But there's no real knowledge about it. Nobody's actually gone through and done any in-depth studies. So even though we have this huge diversity, it's kind of being ignored or put in the back burner. Nobody's looking into it. And first off, what is a trematode, which is what we're studying in the snail? They are internal parasites of mollusks and vertebrates. Most of the trematodes have a very complex life cycle, with at least two hosts, and a primary host where the flukes sexually reproduce is an invertebrate. And the intermediate host, which is an agent of the dispersal, is usually a snail. And I have a better picture later on, but this picture shows the large thing you see is the radia, which is kind of like the mom, I guess you could say. It's asexually reproducing, and you can see inside.
It's the red one.
Oh, thank you. And so there you can kind of see these little black dots like right there. She's full, or it's full, I'm sorry, of the smaller cercaria, which are these. And this happens inside of the snail, this reproduction process. Okay, like here. It starts the definitive host from their waist, the free-living, it's kind of like the eggs. And once they get into the first intermediate host, the snail, that's where the previous picture comes into play. The radia asexually produces the cercaria. And once the cercaria, I guess you can say, been born, they are out free water, they're free living. And then from that they get into other hosts such as fish, crabs, other mollusks, and then another thing eats them like a bird, and so on and so forth. Why did we choose the Black Warrior River? Unique snails are found. There are some unique snails only in this basin, mostly in the Macon National Forest. And so, which is right in here. My original, I'll start this way. Compare parasites of the freshwater at Limiacama among tributaries. Originally, that is what I started out doing. My original project was to go out and actually physically collect, dissect them, and then find which ones carry the parasites. And when I did this, I looked in collecting from here, the Mulberry Fork and Basin, these tributaries, because that's just the area I focused on, mostly because I could get there very easily. I'm from there. And the reason I had to switch projects into a more lab-based study is because the places that were documented years ago to have these snails present no longer did. For different reasons, like this one, it's right up the way from a new coal mine. They had dammed it up, and you can physically see the silt in the water. And these snails do not like the silt. They can't live with it. Another area that was documented a few years ago as having these snails present is obviously dried up. Further up the creek, they had dammed it, so very little water ever gets through. For another reason, they cannot live there anymore. And on. These places were either dried up, dammed up, full of silt from different things being built around it, that these snails, even though there was previous documentation of them being there, have not been found. So due to that, I switched from physically going out to the field and collecting snails to in the lab and analyzing ones that were previously already collected by another individual, and they collected theirs from the Macon National Forest. To do this, the methods was there's isolated parasites from 26 snails that were previously collected from 5 different streams in the Macon National Forest: the Capsea, Thompson, Borden, Brushy, and the Locust Fork. From these samples, 88 parasites were examined individually and measured and photographed. And There's this area, the Bay National Forest, is where all these creeks are present. And once in the lab, the methods and steps used to do this was from each sample, 3 individuals were examined. That means from each sample under a microscope, I extracted 3 individual parasites to take to a different microscope to look closer at them, to get pictures, to get measurements under a dissecting scope, and to determine the larva stage based on tail presence, if there was a tail, if there wasn't one. We used the micropipetter to isolate them. That's what I used to extract the individuals. And then we placed them on the side and looked under at 10 and 40 times magnification using the compound scope so I can look as close as I can to see the different characteristics to get their correct measurements. And under the different magnifications and measurements, the photograph, we use the Moto X imaging software. That software literally connects to the microscope to take pictures through it. And how we consider them different or the same. As we can see on this side, single tail, there's only one tail for each one, and eye spots. You can see this picture, you can clearly see the eye spots. This one you can look closer, but there are right there. And these on this side also have single tail, but there are no eye spots at all. And that's one of the key characteristics to separate them out. There were 4 different types, which later on shows in the table, of actual parasites, but the actual way to separate them visually, the main way is through this: eye spots, no eye spots, single tail or no. Now these are some pictures of them once they're alive. The previous pictures have already been anesthetized, so they're still when I could take measurements. This picture shows that cloudiness, the white cloudiness you can see. That's what they look like to the human naked eye, and you can't actually see them. This just shows what the snails physically look like. It was this type, the Aluminocoma, that we chose to use to see these parasites. And to extract them, we had to, like you can see the top look cut, it's because we had to cut the tips off so they couldn't like curl back up in there so we could get the snail out. And let me see if I can get this to play. This is a video that shows them alive. Just takes just a second. Yep. Now you can see these are the single tail, no eye spots, but see how they move? So because of their movement, sometimes it's hard to take measurements, depending on how they are positioned when they're in this house. Because you can see they curl up and scrunch up and then stretch out to move forward. So you have to be very careful taking measurements to see, are they real scrunched up or is that really how long they are? From the pictures taken, a catalog was created to visually show the differences. This is a snapshot of one page of the catalog. These catalogs are created to show, for future reference, not only the different creeks, but the types of parasites that were found in the snails from those specific locations. So each location had pages showing the different parasites of different types, either with or without eye spots, single-tailed, and so forth. And these are created each time we do studies like this. So for future reference, if we get a snail from like this Tapsy Creek, and we look at this parasite, we're like, well, is this parasite common? Have we seen it before in this specific location? And we can look back at these catalogs that we've created to see if we have or not. And these different types, these are the 4 types of snails that we found. The numbers indicate how many of each one we found. The highest is in 3 and 7, which is what I've already shown with the single tail, eye spots or no. The other ones, they still fall in that category with single tails, with or without, but there's still small differences in them. Unfortunately, at this time, there's not enough research to go on to actually give them individual names. After all of the studies were done, my results was from all the locations, the 5 locations containing single tail, no eye spot parasites. Each one had the single tail, no eye spots. But only one location carried the parasites with the single-tail eye spot parasites. From the 26 snails analyzed, 7 had single-tail with eye spots and 19 had single-tail without. Out of the 88 total parasites individually taken and measured, 23 had single-tail with eye spots, and the remaining 65 had a single-tail without. So obviously from these results, the data is that the single-tail without eye spot parasites are a lot more abundant, a lot more common in this part of the Black Warrior River Basin. From this, the 4 different types of parasites in one species of snail from 5 different streams of Black Warrior Basin suggests that the parasites are very diverse just in these 5 streams. As this is just one portion of one basin, a lot more work needs to be done to understand the trematode diversity in Alabama. As I said before, none of them have specific names. We can't say, oh, that's parasite so-and-so. We just say single-tail eye spots or no eye spots.